Welcome to section 6.3, Cancer and the Cell Cycle. In this section, we'll see how errors in cell cycle control can lead to uncontrolled cell division, the hallmark of cancer. We'll focus on how normal regulatory genes, when mutated, can drive this disease process. By the end of this section, you should be able to explain how cancer arises from uncontrolled cell division, understand the role of proto-oncogenes and how mutations turn them into oncogenes. Describe how tumor suppressor genes act as brakes on the cycle, and explain how mutant tumor suppressors, especially the p53 protein, contribute to cancer. Cancer is not one single disease, but many diseases with a shared mechanism: uncontrolled cell division. Normally, checkpoints regulate when cells grow and divide. When these fail, cells can keep dividing and form tumors. Think of cancer as the breakdown of the cell cycle's built-in controls. Even with checkpoints, errors happen during DNA replication. These mutations can create faulty proteins that regulate the cell cycle. As these mutations accumulate, the cell cycle speeds up or bypasses control points, leading to uncontrolled growth and tumor formation. Cancer usually requires multiple mutations over time, not just one mistake. Proto-oncogenes are normal genes that push the cell cycle forward, like a green light. When mutated, they become oncogenes, which act like a stuck accelerator, forcing cells to divide even when they shouldn't. For example, mutations in CDK family proteins can override checkpoints, allowing damaged cells to keep dividing. Tumor suppressor genes act like the brakes of the cell cycle. They halt division when something is wrong, giving the cell time to repair, or, if necessary, trigger cell death. Important examples include RB1, p53, and p21. Without these safeguards, errors in DNA would be passed on unchecked. When tumor suppressors are mutated, the brakes fail. A key example is p53 at the G1 checkpoint. Normally, p53 can stop the cycle to allow DNA repair or can trigger apoptosis if the damage is too great. If p53 is mutated, cells skip these safety checks, divide anyway, and accumulate further mutations. This loss of function accelerates cancer development. Over half of human tumors have a mutated p53 gene. This shows how critical p53 is to protecting cells. With functional p53, damaged DNA can be repaired or the cell destroyed. With mutated p53, those damaged cells survive and keep dividing, allowing cancer to progress. This is why p53 is often called the guardian of the genome. Cancer is ultimately the result of broken regulation. Cancer develops when cell cycle control is lost. Mutations in proto-oncogenes transform them into oncogenes that drive division. Mutations in tumor suppressor genes remove restraints on division. Together, these changes produce unchecked cell proliferation. Over time, accumulated mutations and loss of regulation lead to tumor growth. Remember, cancer arises not from one error, but from many mutations building up over time.